Nina, while I have you, so what strategies are you employing to address the challenge of pre-existing immunity to AAV vectors in the human population?
Yeah, so this is less of an issue with direct local administration, right? For systemic delivery, though, this is definitely an exciting area of brainstorming. I think we all are aiming to make our therapies accessible to every patient who could benefit. In addition to pre-existing immunity to AAV vectors, we also have to think about reducing the adaptive response, right? Typically with steroids and T and B cell inhibitors. But for the former, one can develop or engineer capsids with reduced immunogenicity, maybe with minimal cross-reactivity to natural serotypes. And then, of course, there are emerging technologies using enzymes, for example, to cleave pre-existing antibodies. I think ultimately, we all hope that in the future, we will discover more approaches to broaden the population that can be treated and make the more strategies even safer. So I think it's really how do we make sure we maximize efficacy with safety in mind.
Yeah, sure. And we're going to get to a whole safety and efficacy portion of our discussion in just a bit. But before we do move on, David, I'd like to talk to you a little bit about the fact. So even on Cell and Gene, we cover rare diseases all, quite frequently. Wondering what, though, from your perspective, what advancements in AAV vector technology do you believe will be the most critical for expanding the application of gene therapy to more common diseases?
No, it's a great question, and it's something that we're seeing. I think, field has kind of gone through these cycles where, it was very much rare disease, and then there was, some more chronic disorders that were being evaluated, and then there was a bit of a look back at rare disease, and now we're seeing, a number of our clients that are working on non-rare, whether they're, like diabetes and heart failure and other things that are much more chronic and millions of patients. So I think it's a new challenge for the field, or a returning challenge for the field. And how do you I think one of the biggest challenges is making it affordable, really bringing down that the cost per dose, and that's not a single thing that to work on. It's back to the engineering of the capsid, so you don't need as much vector to need to administer. And then, from the manufacturing side, more production VGs per liter of production that you're doing. So the overall number of doses per batch are increased. So, I think the field is in a really interesting space where we're seeing higher yields, and we're also seeing move back to, evaluating AAV for chronic diseases. So I think overall it should, I don't know that the amount of production need is going to change, but it's going to be a different application, I think, in the next 5-10 years of going back to really seeing how AAV, based upon learnings that we've seen in the field, how they can now be reapplied back to these chronic diseases.
Yeah, sure, that makes sense. And Nina, I'd like to address the same question to you from your perspective. What do you believe will be the most critical for expanding the application of gene therapy to more common diseases?
Yeah, definitely. So agree with David that there's this huge opportunity for gene therapy and for improved experiences for patients in common diseases. At REGENXBIO, we actually are in pivotal and late-stage development in our retina programs, addressing wet AMD and diabetic retinopathy. These are diseases that impact millions of patients worldwide who are losing vision because of the high treatment burden associated with the current standard of care. And we are on track to be the first one-time treatment here, which is very exciting. Moving forward, improved capsids, as we've already mentioned, in terms of tissue specificity and increased expression and durability will continue to help open up the potential of AAV gene therapy to more diseases.